I will talk about the effect of corundum, surface contamination on aseptic loosening of cementless hip endoprostheses. The research group, Department of Orthopedic Surgery, University Medical Center Ljubljana, MD Medicina Sanatorium Ljubljana, working on clinical orthopedics with MD RI Institute for Materials Research in Medicine, Ljubljana, and Institute of Metals and Technology, Ljubljana, Slovenia. And we run a cooperation on investigations, surface phenomena, wettability, roughness, bone, cell response, surface chemistry, and microstructure for new and retrieved implants of metallic and polymer materials, modification and synthesis of novel surfaces. So, sorry, I will just put this. And with the aim to better understand the premature failure of aseptic loosening of cementless hip endoprostheses. What is our objective is that total hip arthroplasty replacement are the most common and successful methods in advanced surgery to treat degenerative joint disease, for relieving pain, and for correcting deformities. Surgeries have very positive outcomes, but approximately 10% of implants fail prematurely. The most common causes for revision surgery are aseptic loosening and implant infection. So the aim of our work was to study and to investigate the effect of particulate debris from a corundum blasted, that's from this part, titanium implant alloy on premature implant failure, and to find the optimal grid blasted surface. This means that we want to better understand the physical-chemical processes on the rough surface of the femoral component of hip endoprosthesis in connection with the human tissue and liquids is needed for successful bone ingrowth and longevity of the implants. So, aim of investigation, better understanding physical-chemical processes, grid blasted surface of implants in connection with human tissue and liquids. And of course, successful osseointegration, longevity of the implants. So, surface modification methods, the behavior of titanium alloy in the biological system depends on its surface properties. Surface modification to improve biocompatibility during and after manufacturing of titanium alloys have surface contamination oxide layer and plastically deformed outer layer. Such surface is not optimal for biomedical applications, and appropriate surface treatment is the must before its application on any biological system. So mechanical surface modification, that means blasting the surface, is the most widespread process for surface roughness. And this is possible with silicon carbide, with alumina or corundum, biphasic calcium phosphate particles, beta-tricalcium phosphate, and appropriate size is one of the most popular methods for achieving the desirable surface roughness. But for blasting the surface is the most important corundum, white, ultra purity, ultra pure white alumina for medicine. Mechanism of aseptic loosening was represented by Raphael aseptic loosening on this image, we can see bone and implant. And we can see that there is a lot of space here, and a micromotion of the implant relative to the bone, leading to progressive worsening of micromotion, where particles are in the contact between bone and implant here. This was the Raphael, how to say, mechanism, and also that this, how to say, were particles activating macrophages, initiating a cytokine signaling cascade that leads to osteoclastogenesis and bone resorption. But Doliner and I found that also corundum particles are the cause of, how to say, aseptic loosening, besides other wear particles from corrosion products and also from polyethylene particles. We have different protheses for total hip arthroplasty, and this is ceramics to ceramics, which is this one is ceramic acetabular ball and a ceramic acetabular cup. And we have metal to metal, which is now, how to say, so-called forbidden because of cobalt metallosis, which comes from cobalt chromium materials, and cross-linked polyethylene with ceramics is this one. This is cross-linked polyethylene and ceramics. So this cup is here, and how to say, metal to such cross-linked polyethylene and ceramic to polyethylene and metal to polyethylene. Acetabular and femoral components are different. Fixation is possible cemented or cementless. In our case, we work with cementless. Surgical approach is possible to anterolateral, posterolateral, and anterior. And here we have on this image, we have, how to say, schematic of cemented hip endoprosthesis. So in other cases, there is no cement, but here is cement, which is only for older patient with very bad bones. So here we have again the picture or photo image of different bearing, and of course wear rate, which is quite enormous for this cobalt chromium and ultra high weight molecular polyethylene, but it is very low wear rate for ceramics to ceramics. When we put such a endoprosthesis, hip endoprosthesis into the body, there is a very important biointerface between biomaterial and biosystem. And here is very important what are the most important things on nano, micro, and macro level. On nano level, it's important that we have metal and oxide. In micro are crystallites, grain boundaries, and surface roughness. And how to say, geometrical design on the macro level. Here is an example for dental implant, not for, how to say, hip implant, but situation is the same. The most important is this biointerface. On the other side, we have proteins, cell membranes, cells, and of course, tissue. And we must, how to say, control everything, not only biomaterials or tissue. Materials and methods, we have retrieved titanium, alumina, niobium, 2 Miller femoral stems of cementless hip endoprostheses, selected from a register and repository of University Medical Center in Ljubljana. And it was about 2013 2 Miller. Then we have studies of selected hip endoprostheses, premature failed due to aseptic loosening. It was 10 pieces infection, latent infection, and we have two new for comparison. Methods where multivariate analytical methods were used, contact angle, water measurement, and profilometry for hydrophilicity, wetting properties, and roughness. This is a Licona device. Then we use scanning electron microscopy, a field emission scanning electron microscopy, size Gemini, crossbeam 550 with integrated techniques, electron spectroscopy techniques like energy X-ray dispersive spectroscopy and electron backscatter diffraction were used to examine surface, chemical composition, morphology, phase analysis, and of course, microstructure. Then we use X-ray photoelectron spectroscopy for surface analysis and time-of-flight secondary ion mass spectrometry of thin oxide film on the rough surface. But in the fact, we can measure or study this passive film only on polished surface. It is not possible to use so sensitive methods like X-ray photoelectron and time-of-flight films for analysis of such a rough surface. So here on the first image, we have secondary electron image of morphology of the surface. Then this is a new sample, and here I mean a new stem was not used. And here we have red is matrix. This is titanium alloy with 6% of alumina and 7% of niobium. And the blue is Al2O3 or aluminium oxide or alumina, which is the retained corundum. This was already, how to say, known, but it was not known that this corundum is also into the subsurface. Up to the 20 microns of subsurface, we can find contamination of corundum. And here is EDS, electron dispersive X-ray analysis, and in this case is mapping. For that reason, we have different colors, and this green is again this corundum, and red is matrix. And here we have also some spectrum, EDS spectrum. So retrieved implant premature fate of infection is sample one. Here are only cross sections. And we see also, how to say, corundum here, and a lot of cracks because this corundum is very hard material, while the matrix is softer. And for that reason, because this grid blasting of the surface is not how to say machine, but it is by hand, and for that reason is not homogeneous. We have this. Then here we have again, and we see such cracks, and this how to say particles can release, can be released to the soft tissue near the hip. And then we tried to find, to prepare our oven at ferroturtolite, which is very known, world-known producer of these devices for industry, and here we get the same results. And there are XPS time-of-flight sims, analysis of this, how to say, sim oxide film. On the first image, we can see this XPS depth profile of a new titan aluminum niobium alloy covered with native oxide after polishing. Thickness of oxide was estimated to be 6 plus minus 2 nanometers, and XPS depth profile of retrieved, this one is retrieved, is a little less. The thickness of the oxide is 5 plus minus 2. And here we have also a figure of seams depth profile of native oxide layer on the polished surface on the new titanium hip implant. Thickness of the oxide layer is about 6 nanometers, what is in accordance with XPS analysis. The oxide layer consists of mixed oxide, titanium 2 dioxide, like layer is on the top. Beneath is aluminic oxide layer. At the interface with the matrix is titanium O suboxide layer. In the oxide layer, also niobium is present. Then we made the first, how to say, assessment of cytotoxicity using human bone marrow derived mesenchymal stromal cells in direct contact systems according to international standard for this, how to say, testing. Materials which were we tested was corundum ceramic, white fused alumina discs, then polished corundum fine particles of granulation 300 to 425 microns, titanium block sample rough surface blasted covered with corundum, and titanium block sample polished. All materials were sterile, packaged separately. The materials were tested with the human primary mesenchymal stromal stem cells culture derived from bone marrow in a direct contact system where the biomaterials is in direct contact with the cells. The biomaterial were fixed on the bottom of the cell culture vessel, and primary human cells were seeded evenly throughout the cell culture vessel, including biomaterial surface in cell culture media. The biomaterial cells were incubated on 37 degrees centigrade, 5% of CO2 for 7 days. The cells in the vicinity of biomaterials were monitored for their morphology, and after 7 days, the biomaterials were stamped with inspected for the presence of live cells. The controls were also. And here we can see that the results of daily monitoring, cytotoxicity testing. The photos represent the cells grown in immediate vicinity of the materials. Ceramic discs show no sign of cytotoxicity. Corundum small particles exhibit most obvious cytotoxic effect. With titanium samples, one small level of cytotoxicity was detected only where corundum cover was present. When those samples were polished, there was no cytotoxicity effect. So for conclusion, we found corundum particles contamination on the grid blasted surface of new implants of 3 different worldwide recognized manufacturers. And retrieved implants premature failed due to aseptic loosening, infection, and latent infection. The residues are potential problem, third body wear induction for osteolysis and aseptic loosening. We first discovered the subsurface corundum contamination of all investigated and new and retrieved titanium alloy stems for 5 down to 20 microns. We found cracks near the corundum particles in the subsurface and could be adhered also by bacteria, causing also implant infection. And preliminary investigation shows that the retained corundum decelerate osteointegration. Thank you for your attention.